Right. Thank you very much, Hira, for the introduction. Very nice. And also very grateful to Sarah Spalding, who invited me to give this seminar. And I hope that you can learn something about the diatoms in the plankton in the Mexican Pacific, and also enjoy some of the photographs and the images that I am going to provide in this seminar. Well, okay, next. Okay, I will start this by using this paragraph by Michael Wyrie. The last year, he provided this revision of the number of species of algae, and it was amazing to confirm that diatoms are very diverse, with more than 18,000 species. More of these species were living, more than 16,000 living species of diatoms. So this gives a very special account of the diversity of diatoms from freshwater and marine environments. I'm trying to focus on this seminar. I will say that the littorals of the Mexican Pacific Ocean are considerable in length and comprise various areas and diverse environmental conditions, from tropical to subtropical in the southern part to more temperate in the coast of Baja California and Baja California Sur, and includes two main gulfs, the Gulf of California and the Gulf of Tehuantepec in the southern part. We have been carrying out some oceanographic cruises, especially in the area of the central Mexican Pacific, which goes from Cabo Corrientes to Acapulco. That's in the panel of the right side. We have gathered lots of data information about marine phytoplankton, but especially marine planktonic diatoms in this area. The materials I'm going to show were obtained from field samples with both net and bottle in different, many oceanographic cruises. And also we have established different cultures of planktonic diatoms. And we have been trying to understand the diversity through this metabarcoding approach along coast of the Mexican Pacific. So we have been studying the diatoms with a microscope or different microscopes, and also using molecular tools. And we have lots of different environmental measurements when we use the ships for oceanographic cruises. About 4 years ago, we published this sort of catalog of phytoplankton of the central Mexican Pacific, which includes dinoflagellates, diatoms, and some other phytoplankton species. And it includes 501 species identified and illustrated, with 257 dinoflagellates. Surprisingly, it was the taxonomic group with more taxa identified, and the second group was diatoms. Of course, my appreciation for diatoms led me to suppose that diatoms were the most diverse group, but in fact, dinoflagellates were, in this book at least, more diverse than diatoms. Among the diatoms, the diatom genera most rich, species rich, were Chaetoceros, Thalassiosira, and Rhizosolenia. Right, this is to show some of the content of the book, including some illustrations of species of Asteromphalus in light microscope and also scanning electron microscopy, several species of Asteromphalus. And then another centric diatom, Thalassiosira or Thalassiosira, with different species illustrated again by light microscope and also scanning electron micrographs. Then this very important genus in the plankton, Chaetoceros, with different species illustrated. And then Pseudo-nitzschia, which is also important because of the some of the species of Pseudo-nitzschia, they may produce some a toxin, domoic acid. So they may be very important. Fragilariopsis as well, forming chains. Okay. I am going to give you some more examples of the diversity of marine planktonic species in the Mexican Pacific with different genera, Minidiscus, Mediolabrus, Thalassiosira, Skeletonema, Actinopithecus, Chaetoceros, Nanoneis, Thalassionemataceae, Lioloma-nitzschia, and Pseudo-nitzschia. Most of the species were studied from field samples, net or bottle, and some of the bottle samples were filtered to have this, the material on filters. And this is especially important for very small species like Minidiscus and Mediolabrus. Minidiscus is the first example with many species found, including this Minidiscus trioculatus, and then Minidiscus chilensis. You can see different morphological characteristics of this species. And then a new species that we are going to propose, and we are going to give the name of Baja Californianus, because it was found in the coast of Baja California, in the western coast of Baja California in Mexico. It is very small, as you can see. The central area is hyaline, and also the marginal area has no areola, is also hyaline. And you can see the size is very, very, very small. Some more details of this Minidiscus is given here with some views of the valvocopula, copula, and pleura, including the small poroids in there with the arrows. And no rimoportula, only fultoportula in the central area, as you can see in the figure down there. Next genus is Mediolabrus, which is represented by Mediolabrus comicus, described by Takano in Japan. It is present in the Mexican Pacific. Mediolabrus spinulosus, which is a new record in the Mexican Pacific Ocean, described originally in China. And this species, Thalassiosira delicatula, was established in cultures, so we could see some details, this high variability in morphology, including the presence of one or two central fultoportulae. You can see one in the figure 11, and in number 12, you can see a couple of these central fultoportula. Chain-forming species, Thalassiosira delicatula, and another Thalassiosira species, Aleni, also cultured, producing some auxospores, but also giving us the some details of the morphology of the valve, the different processes, including the photoportula, rhemoportula. The rhemoportula in this species is prominent, as you can see in figure 18, or in this figure 17 as well. The difference of the external extensions of these processes. And more cultured species of planktonic diatoms include Skeletonema tropicum, Skeletonema grevillei, Skeletonema pseudocostatum, and we didn't find the original Skeletonema costatum, which was supposed to appear in all the waters of the Mexican Pacific, but in fact, now we know that Skeletonema is a complex of different species, and these 3 species were cultured for one single bottle sample in the mouth of the Gulf of California. So we can also add this Skeletonema ardens. These 4 species of Skeletonema were found in just one single bottle sample, and they were successfully cultured, and it gives the impression of the big diversity of different genera of diatoms. Skeletonema ardens is like this, very small, and also we could manage to get Actinopithecus scenarios in culture with the development of auxospora as well, and the formation of chains. You can see in this figure forming chains, curved chains as well, and they were joined by this very short mucilage thread. So it is very exciting to see this Actinopithecus scenarios in culture. Now we are going to the Chaetoceros world. Chaetoceros, as I told you, is the very species-rich genus, and we have recognized some of the new species, including this Chaetoceros crawfordi. This was dedicated to Richard Crawford, who was my supervisors for PhD in the University of Bristol. And this is a chain-forming species, very long. And you can see that it is joined not only by the setae, which are fused, but also these small bridges in figure number 8 and 9. You can see the bridges, which are joined by silica. And in figure number 10, you can see the elevations of these structures here in separating chains. Yes, this is also shown in figure 11. And we are still studying the diversity of Chaetoceros, including this very bizarre species of Chaetoceros splendidus, very, very strange, with the intercalary setae joined for some time and very thick, including spines. And also these cells of the chain are joined not only by the fusion of setae, but also the fusion of the central part of the valve face. Another species which is new is this Chaetoceros species number 2. The formation of the terminal setae is very characteristic to setae. Let's say the setae are thin, but one setae is very curved, different from the other one. And these terminal setae are different from the intercalary setae as well. And another species which is considered to be new is this Chaetoceros species 3, with only 3 cells in the chain. I can tell you that the city have the same structures as of the species Chaetoceros bacteriastroides. So it may be related to that species, but it is completely new. Now we are going into the pinnate diatoms, including Nanoneis. And Nanoneis, originally Nanoneis haslea was described some time ago by Norris for very small forms with only a kind of rave. It's not completely rave, it's just one part, and it is termed rave, but in fact we don't know if it is a real rave or it's another structure. And they form small or short chains with 2 or 3 cells attached with mucilage. Yes, so this is the very small Nanoneis haslea. You can see the magnification and the scale. This species is very small. And it was separated from Nanoneis longta by Zhang Li in China. And the main characters separating these 2 species were that Nanoneis longta is indeed longer than Nanoneis haslea, but the formation of chains is about the same. And the number of characters probably does not allow to separate both species. We have found some intergrades of Nanoneis haslea and Nanoneis longta. And yet we found another Nanoneis. By the way, I will return to this to show you one characteristic of this morphological characteristic of this species. The margin has this porticus marginalis, which is a raised structure, having this like portals, these structures. So this porticus marginalis is a characteristic of Nanoneis haslea and Nanoneis longta, but it is lacking in Nanoneis californica, which is also very similar in length to Nanoneis longta. But if you see carefully, the marginal porticus marginalis is lacking, and instead we can find these structures like small undulations here along the margin of the valves. So we are trying to define the new species Nanoneis californica. And then we are going to Thalassionema and the very common species Thalassionema nitzschioides, which was originally described in the Gulf of California. It is a species forming chains in zigzag or stellate chains. The morphological variability is considerable and includes the variety parvum, which is a very small form of Thalassionema nitzschioides. You can see the size in the figures 43 and 44 compared with the size of the given taxon Thalassionema nitzschioides. Thalassionema kuroshioensis was described originally in Japan, and we found this also in the waters of the Mexican Pacific. It is a solitary species with 2 poles very similar, no difference, and double-lipped rhimoportula. So this is also present in the Mexican Pacific waters. And also we found Thalassiothrix patulata, which is a more tropical species. And this has 2 different poles. You can see the figure number 74 and 75, 2 different poles. One is is wider and more similar to spatula, so that the name comes from there, spatulata. And important characteristic is that they possess these very pointed spines in the margin of the bulbs. You can see lots of spines in figure 74 and 81. Both poles have different rhimoportula. You can see 84 is more or less prominent, but 86 and 87, they have dissimilar lips of the rhimoportula. So these both poles are different. So one is called foot pole and the other is head pole. Another genera is Lioloma, and the name designates some species with no spines like Thalassiotrix. So it has no spines, but it has two different poles as well. And also we can find these bubble-like structures in figure 103 with the arrow. You can see the bubble-like structures. We don't know the functions. We are speculating the possible function that can have these structures for keeping buoyancy, perhaps. That's one possibility. The rhimoportula, they have this dissimilar lips. They have this parrot beak shape, as you can see in figure 105 and also 107. So the lips are dissimilar. So we are approaching to the Pennids diatoms with a rave, including Nitzschia leyi, which is a very, very small species, very common in tropical and subtropical environments. It can be important in densities, but it is very small, and sometimes it is very difficult to separate from other species like Nitzschia bifurcata and some others like bicapitata and other more. You can see Nitzschia leyi is this with this morphological variability and the number of gill bands, 4 bands here in letter D. But in the panel down, you can see Nitzschia bifurcata and some other related species. And sometimes it is very difficult to separate all these species, and you have to rely on the images of the electron microscope to separate or to see some very small characteristics like the bifurcate stria. In bifurcata, the stria originally can be just one stria per one row of poroids per stria, and at the end, this single row become double with double rows of poroids. So you can see the variability of different species of Nitzschia, which are common in the tropical and subtropical waters of the Mexican Pacific Ocean. These species were cultured from samples in the Mexican Pacific, including some of the toxic species of Pseudo-nitzschia. Pseudo-nitzschia brasiliana was considered not to be toxic, but in fact, our student found some production of domoic acid in Pseudo-nitzschia brasiliana, whereas in Pseudo-nitzschia pungens, it is known that it produces some of this domoic acid. They are not so easy to identify, but if you use some transmission electron microscope, you can find some differences which are important to separate these species. Pseudo-nitzschia multistriata is the second species producing more domoic acid per cell, and it is also present in this area. You can see the very fine poroids of the stria in this figure, down in this panel. Right. We also found Pseudo-nitzschia cuspidata with some details of the row of big poroids in the stria, with 2-3 different sectors. So it is also considered to be toxic, this Pseudo-nitzschia cuspidata. And some time ago, we described a new species of Pseudo-nitzschia in the Gulf of Tehuantepec. This was dedicated to Professor Frank Round in the University of Bristol. And it is characteristic for the shape, but also the number of poroids, one single row of poroids per stria, and the shape. It is related to Pseudo-nitzschia subpacifica, but Pseudo-nitzschia subpacifica has 2 rows of poroids. 2 new species were also described not from the Planktonic Diatoms, but also from the epipsamon, this environment with in the sediments. We found 2 different species, Phyxeliella pacifica, new species, and Cerataulus simsae, dedicated to Pat Sims. Right. This is the approach with the metabarcoding analysis, and you can see that from the different phyla, which are included here in Protoctista, Arthropoda, Cnidaria, etc. Bacillariophyta was the 4th most important in terms of abundance. Also, this allowed us to separate some potentially toxic species of Pseudo-nitzschia, which are encircled in this area: Pseudo-nitzschia seriata, pungens, fraudulenta, cuspidata, australis. So you can see that in a number of different stations, they are present following this metabarcoding approach. Also, in this other slide, you can see that diatoms are important through this long list of different taxa orders here. And Chaetoceros was the most abundant taxa among the orders here, while centric diatoms were the most important in terms of phytoplankton classification. Also, in this slide, you can see the presence of this species which are considered to be potentially toxic of Pseudo-nitzschia. I am showing this map to you to see where these stations are situated, more to the south in Acapulco or to the north in Cabo Corrientes. And then they are grouped, these stations are grouped by the similarity, with the more coastal stations to the left side, and the more oceanic stations to the right side. They were having more or less the same communities with different dinoflagellates and diatoms, potentially toxic. Right. Along this study, we found different relationships, symbiotic relationships with Dactylosolen and Solenicola symbiosis. You can see the diatom. It's completely covered by this Solenicola. See, this is more dramatic. So we found this, but also this very strange combination of Thalassionema, nitzschioides, joining Thalassiosira in this special symbiosis. We don't know if this is just a miracle or something, but this Thalassionema is attached to this to the Thalassiosira. So this is very strange. The other symbiotic relationship is Thalassiosira with Reticulofenestra, which is a coccolithophorid. So this coccolithophorid is attached to the girdle bands in Thalassiosira punctifera. So we don't know what is the real association between these two species. I suppose that this Reticulofenestra is very heavy, so the diatom is very limited to remain in certain layers of the water column. Despite of that, one of my students found that this relationship has densities, considerable densities in Acapulco. So we don't know if this is important in terms of buoyancy or not. Right. Additionally, another student made some analysis of domoic acid, the toxin produced by some species of Pseudo-nitzschia in the central Mexican Pacific, and the concentrations were relatively high, from 40 to 690 nanograms per liter in these different stations. You can see the table here with the concentrations. Station A31 was having this concentration, the most important concentration, with 0.669 nanograms per milliliter. This is associated, of course, to the environmental characteristics of the water column, and you can see this in this slide showing the maximum chlorophyll layer here, which is not exactly in surface, but in subsurface, around 20 to 30 meters depth of the water column. Right, some of the conclusions or important points to be discussed is this symbiotic relationships were detected, and we are trying to discuss the what is the meaning of all these very strange relationships. Many of them have been documented previously, but some of them are new to us. The analysis of domoic acid was also performed. We know that this toxin is present in the Mexican Pacific, and we can say that the distribution and certain ecological patterns of planktonic marine diatoms have been detected in the Mexican Pacific. For instance, in one single sample, up to 4 different species of Skeletonema may coexist in a single spot or in a single sample, which is very important to us. And about distribution, this species, Thalassionema basilaris, Inedriforme, Thalassiotrix giberula, and Thalassiotrix spatula, were more restricted to tropical and subtropical areas. They were not found in more temperate areas. Well, we can confirm this. And about Pseudo-nitzschia, the high densities of the some species are associated to particular climatic, oceanographic, and hydrographic conditions, including El Niño, La Niña, or upwellings, turbulence, stratifications, nutrients supply, And the subsurface height densities were common, as it was shown in the previous slide. And of course, we have to study many aspects about the diversity and the ecology of planktonic diatoms in the Mexican Pacific. Right, I hope that this has been important to you. So I thank you very much for your attention, and I am giving gracias, many thanks to you for your attention to this seminar. Many thanks.
Thank you, David, for that really amazing seminar about the incredible diversity. It looks to me like I'm entering another world of these diatoms that are so foreign to me. I have a lot of questions, but I want to give people a chance. Tom, I see you here, and I'm looking forward to what the marine folks, how this discussion develops. Maybe what I'll start with, Tom, you're going to recognize this. I'm just going to try this mystery diatom out on David that we've been tossing around from someone on the, maybe someone on the seminar has an idea what this thing might be. Can you see that, David? We don't even know if it's a diatom. It sort of, we've talked through.
Oh yes, oh yes. Wow, wow, wow. Wow, wow, it's wonderful.
Yeah. Anyway, anybody who wants to pipe up and comment on that. Matt, I see you've got your hand raised. Why don't you go ahead?
That's lovely. Well, thank you very much. Yes.
What happened to Matt? Oh, there you go. Okay.
I'm here. I'm here. Sorry, getting caught up with all the button pushing. Thanks, David. That was a really, really, really fascinating talk. And like Sarah said, a lot of questions. So I'm going to try to limit it to one here, and I'll pester you with emails for the rest of the day. But with regard to the Actinopithecus that you had in culture, and you said there was an oxospore. Do you have any idea if that was a sexual oxospores? Were you seeing gametes in the cultures?
Aha. Well, that's a very good question, Matt. This is not the first time that we can see some oxospores produced by different centric diatoms, including Thalassiosira, Thalassiosira, and Actinopithecus, and some others, Chaetoceros as well. In fact, we have not seen these gametes or the masculine gametes, which are supposed to have a one flagellum. So I suspect that sometimes, or most of the times, these oxospores are produced without sexual productions of gametes or something. Of course, sometimes it's difficult to make sure that you didn't see any of these structures. But I mean, if they are supposed to be flagellate gametus, the masculine gametus, so it should be possible to see something swimming around. So, and we didn't see anything. So we are proposing one of the papers that sometimes these oxospores may be produced without sexual reproduction, which is
yeah, I think that happens a lot more than we think so. So yeah, that's what I was
inside in that. Yes. I thought it was a kind of the different, product of our imagination or something, but they are there, and we couldn't document any of these gametes.
All right. Thanks.
Yes. So anyway, to try to find something about that to make some conclusions, but in different cultures we couldn't find these gametes. Yeah.
David, I was really fascinated by the examples of symbiotic relationships.
Oh yes.
And what you mentioned something called Selenicola. Solenicola?
Solenicola.
Solenicola. What is
Solenicola.
what type of organism is that?
Yes, as far as I know, this Solenicola is part of this group of protists called straminophiles.
Okay.
In the name should be something like mast, or related to the mast, which are marine straminophiles. And but some other authors, they have said that they are kind of fungus, and some straminophiles are fungus in kind of fungus as well. So it's difficult to at the moment I cannot say more about that, but there are some 2-3 papers given by some other authors, including Fernando Gomez. We could need to have some of the molecular sequences perhaps to make sure which is the
and also the relationship you
Solenicola.
showed with the coccolithophorae on the Thalassiosira. Has this been widely reported? Is this something that is well known in the marine world about these kinds of associations?
You mean the Thalassiosira with the reticulofenestra? No.
Okay.
No, with the Thalassionemataceae, yes?
Well, all of them really, just the degree of whether they're, associations, symbiotic or
Well,
mutualist
I mean,
or
I
antagonistic.
suppose that diatoms may be attached to a different substrates, let's say. If this is another diatom, could be kind of accidental situation, but I don't think this is the more usual condition. Sometimes you can find this accidentally, and it's difficult to explain, but this is an exception indeed.
Okay.
It's difficult to give more explanation without knowing which is the function of different chains. For instance, we suppose that the chains are produced to get together the cells in the environment. The environment is very turbulent sometimes, but we don't know truly which is the function of chain forming diatoms. So yes, it's difficult to know.
And you showed a very what appeared to be, if this is a stable layer at 20 to 30 meters deep, where these forms are maintaining their buoyancy. Is that the understanding, that they are able to persist at the depth that they?
Yes. Well, my experience, which is for some years now, working on marine phytoplankton, I have learned many things about the some special characteristics of the planktonic community. Buoyancy, of course, avoiding predators, and trying to find some affinity with other communities or species related, for instance. But in fact, it is very difficult to go into the ecology in deep, especially if you need to measure different let's say parameters or variables, or yes, and you probably will need to have special equipments for very microscopic things like this. So yes.
We've got some questions in the chat. Layla says, thank you for your talk. What about Rhizosolenia and its diversity? Have you noticed an obvious change in diversity between subsurface and SCM? I'm not sure what SCM is.
Rhizosolenia, about buoyancy, or?
Yeah, Layla, maybe you can clarify that if you want to go ahead and and bring your question up. Thank you.
Hello, thank you for your talk. I wondered about the diversity in Rhizosolenia because I saw it quite different from the Bering Sea to the pole, and with association with some other species, but it wasn't always the same in subsurface sediment samples and in the maximum chlorophyll SAM profounder samples.
Right.
So I wondered if you noticed the same.
Yes, well, this point was discussed previously by some other authors like Tracy Villarreal some time ago about buoyancy and this position of the Rhizosolenia, the very big ones in the water column in tropical areas. I don't know much about the Rhizosolenia in more in polar areas or in yes, Antarctic or Arctic. I don't know what is the situation, but I know that Rhizosolenia may be very they can have high densities. Yes.
But in your site, you talk as well about Rhizosolenia. So where have you found it?
Well, yes. They are distributed along the water column. Not we don't know any special information about the precise location in the water column. We have found also this symbiotic relationship with this cyanobacteria, Rickelia intracellularis, which is also supposed to be a more tropical relationship. And this cyanobacteria can fix atmospheric nitrogen. So this is some kind of, symbiosis very explicit, let's say. I also found this Rhizosolenia with the species Silroteca closterium inside a Rhizosolenia. Living samples, the Silroteca was living inside a Rhizosolenia. So that's another situation which we cannot explain at the moment. But about the distribution in the water column, I cannot tell you something more information about that. I'm sorry.
Okay, thank you very much.
Thank you. And Anna also has a question along the lines of what I was asking about the coccolithophores on diatoms. First, is it widespread in the population that you observed, and then or across populations spatially?
Wow, that's very difficult to answer. I don't know. We have different questions about the how is it possible for some coccolithophores to attach to the diatom, but and about spread or something. I have
could they be attaching on mucilage? Is it a
Oh, well, yes, that's possible. Yes. I am trying to think about this very close genus Planktonella. Planktonella may produce a kind of wing in this area in the girdle bands, and mucilage is probably the material for producing this wing in Planktonella sol. I don't know if this mucilage can be produced by the diatom, the Thalassiosira, to allow this coccolithophore to get attached to the diatom. So yes, there are many questions about the attachments and relationships.
Yeah, I know that Shinya Sata in Japan, he and his student are doing some very interesting work on bacterial associations within the diatom mucilage.
All right.
Exterior cells, and some that appear to be promote growth, and then some that are antagonistic. So yeah, I think this is another area that we don't really know much about, and it could be very
yes.
It makes me think too about what are the with domoic acid, what role is it playing? Is it having things that are microbial?
Yes, to study all these aspects of ecology and physiology, and yeah, different questions we have.
Yeah.
So we have some more questions from.
Yeah, here, Tom, go ahead.
Frankovich, yes.
Okay. David, thank you for coming. It's a real honor to have you on diatoms.org. And if I can, extend an invitation to you and your lab to produce taxon pages for diatoms.org, it'd be a great, it would great to have our neighbors across the Gulf of Mexico on here. And we really appreciate you being here. So I have a couple questions. One is real simple, and I have to apologize. That green line you saw, and that was me. I was trying to copy a link to the catalog, which didn't work, so I shouldn't touch things. But how can we get a PDF copy of that catalog that you referenced early in your talk? We'd like to get that.
Yes, that's the first question. Yes, well, that's not very easy because this book was produced by my own institute, and the distribution is very limited. So most of the time, when I visit some labs or go to different scientific events, I get some books to distribute to among some of the colleagues. But if you can provide your address, probably I can send you a copy of this book. If it is necessary for you, I can just send it to you.
Yes, thank you very much. And yes. I have a more general question. After many decades of working with marine planktonic diatoms, are we any closer to saying why do we have all of a sudden an abundance of Chaetoceros, and then a month later we got an abundance of Thalassionemataceae, and then an abundance of Thalassiosira? Can we tell anything about the niches of these groups?
Well, it's absolutely impossible to answer this question with certainty. But if you, I mean, we have been culturing some diatoms, and we now know now that some diatoms, they grow very fast in cultures like Skeletonema, Thalassiosira, and Chaetoceros. So they are kind of opportunistic species, of course. So they develop some very high densities in very short time in cultures. So if they don't are afraid of the grazing of copepods or some other predators, they can grow with no problem to I mean, grow and grow or grow. If they have the enough amount of nutrients. So they just are machines of growing, especially this genera. Of course, some other genera, for instance, Minidiscus, they grow slower. That's for sure. But Skeletonema and Thalassiosira, wow, they are growing very, very fast. So they are kind of opportunistic, and if they don't have any special limitations, they can grow, in time and in space for some time.
Okay, thank you. I really enjoyed seeing the diversity and
well,
sort of in person.
Yes. we have to understand many of these situations in the ecology of not only in diatoms, but in some other phytoplanktons.
Yes. Yes. Thank you.
Well, thank you very much. Yes.
Well, we're already past the top of the hour, and I wish we could go on because I still would like to carry this conversation on. But I really want to thank you again, David. It's been wonderful.
Well, thank you.
and
Thank
enriching
you.
for
And,
this group.
well, I
And
was happy to show some of the work that we have been doing for some time with students and colleagues. Of course, my name appears in the seminar, but behind there are students and colleagues that have been contributed to the understanding of this subject. Yes. Okay. Well, thank you very much.
yeah, so thank you. And I'd like to thank everyone who joined today. You all make this really such an enriching discussion and environment. I really appreciate you being here. So thanks so much. And we'll see everyone for our next Diatom Web Academy in 2 weeks. So be well and stay in touch. And thanks again, everyone.
Well, thank you very much.